Hello and welcome to Nikolai's genetics lessons, and today's video is going to be about population genetics. And here's a problem: you're conducting a study of an isolated tribe in New Guinea, and you find that there is a widespread resistance to cryptosporingium infection. You determine that the gene for resistance is inherited in a recessive fashion. The incidence of resistance in a normal population is 1 out of 900. In New Guinea it is 1 out of 25. What are the carrier frequencies in the normal population and in New Guinea, respectively? Assume that the populations are in Hardy-Weinberg equilibrium. And here is a variance of the answers. As usual, I recommend you to pause the video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. In order to solve this problem, all you have to know is just Hardy-Weinberg formula, which tells us that frequency of the three genotypes, which can be produced by two alleles in a population, would equal to p squared plus 2pq plus q squared. And all these three genotypes, when we combine them, we should get 1 or 100%, because 1 in our calculations would equal to 100%. In statistics, we usually do all our calculations on the scale between 0 and 1, so 1 equals to 100%, and for example 0.5 would equal to 50%. In statistics, we use such symbols as P and Q, but you probably get used to symbols like dominant A and recessive A. So for example, p squared would stand for the genotype which is homozygous dominant, 2pq would stand for the genotype which is heterozygous, and q squared stand for the genotype which is homozygous recessive. As you see, those we have three genotypes here, but these three genotypes are made by only two alleles, p and q, or dominant allele A and recessive allele A. So all the alleles p plus all the alleles q in this population equal to 1 or to 100. So p stands for the dominant allele A and q stands for the recessive allele A. Now let's read our problem again. You determine that the gene for the resistance is inherited in the recessive fashion. So resistance is due to recessive allele, and person who is homozygous recessive would be resistant. The incidence of the resistance in a normal population is 1 out of 900. 1 out of 900 people would belong to this genotype and would be resistant. So let's write down 1 out of 900. So what this tells us? This tells us the frequency of the homozygous recessive genotype. But in order to solve this problem, how many heterozygotes we have, we need a frequency of the recessive allele and dominant allele. So take a look. Q squared equals to 1 out of 900. Then Q would equal to, Q would equal to square root of 1 out of 900. So Q would equal to 1 out of 30. What we have just found, take a look. Q squared stands for the homozygous recessive genotype, and Q stands for the frequency of the recessive allele. If we now know that frequency of the recessive allele is 1 out of 30, and we can tell the frequency of the p allele. So it's going to be 29 out of 30. So if we combine these two numbers, we are going to get 30 over 30. 30 divided by 30 is going to be 1. Again, this is frequency of the dominant allele, and this is frequency of the recessive allele in general population. Now, in order to answer our question, and our question is what are the carrier frequencies in normal population. This question about the frequency of this genotype, which is 2pq, and this is heterozygous genotype frequency of the carriers. 2pq, which stands for the frequency of the heterozygous genotype or carriers, would equal to 2 times 29/30 and times 130. And the answer is going to be 2 times 29 is going to be 58. So 58/900. And this number stands for the frequency of the heterozygous genotype in a general population. Now, second part of the question, and what is the frequency of the carriers in New Guinea? People in New Guinea were under pressure of this disease, so of course we understand that they would have more people who belong to this genotype, which is resistant to this disease, and of course also would have more carriers, because frequency of the recessive allele in the population would be much higher. Now I'm going to use different color in order to solve second part of our question. And we are told that the frequency of the people who are resistant to this disease is 1 out of 25. So 1 out of 25 people would be resistant, and that means belong to this genotype. Small a in our Hardy-Weinberg formula, it is stands for the q squared. If q squared equals to 1/25, and q squared stands for the homozygous recessive genotype, then in order to find the frequency of the q or recessive allele A in this population, we just have to take a square root of 1/25. And the answer is going to be 1/5. Now let's return to this part. If we know that the frequency of the recessive allele is 1/5, then we can say that frequency of the dominant allele, P, would be 4/5. So if we add 4/5 plus 1/5, we are going to get 1 or 100%. So dominant allele plus recessive allele, if we combine them, we will get 100%. Of course, we are going to get 100% because we have only two alleles, so they would represent 100%. And now our last step is going to be to find frequency of the heterozygous genotype. So again, this part of the formula, which is 2pq, and we have 2 times 4/5 and times 1/5. And in this case, we are going to get 8/25, and this is going to be an answer to the second part of the question. So here is the answer to the first part of the question, and many answers has this number, but answer to the second part of the question give us only one choice, and this is going to be answer D. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.